I'm happy to be here to be able to talk to you about hollow fiber bioreactors, and also hopefully we'll come away with a little better understanding of standard flask culture and some of its limitations and factors that affect your research. Hollow fiber is not a new technique. It's actually been around since 1972. It was developed by Richard Nazik at the NIH, and he was looking to develop a more in vivo-like way, a more three-dimensional way to culture cells. It's really only been in the last few years that we've learned about what the real power of hollow fiber bioreactors can deliver, and the differences in terms of how cells behave in their physiology when cultured in 3 dimensions at high density in a hollow fiber bioreactor. So we can kind of start with the Michener approach to cell culture and harken back to 1906 and Harrison, who was the first to attempt to culture cells in vitro. And he was actually utilizing frog neuronal explants on plasma clots, a 3-dimensional matrix for culturing cells. Through the years, we've gone from using glass flasks and now to plastic flasks. It's actually relatively new technology. I was in graduate school in 1980 and don't recall too many laminar flow hoods or incubators or cell culture being performed in the laboratories around me. It really took, historically the development of digestive enzymes from Worthington in the late 40s and early 50s to do tissues dissociation, the development of cell lines in the late 50s and early 60s. It was a very rigorous and dogmatic technique until we had laminar flow hoods developed, which were in the mid-70s. So in the historical sense, it's really quite surprising to me how ubiquitous cell culture is as a research tool and how relatively recent the technology is. So we moved from flasks to roller bottles. Biology is all about cell culture and surface area when we want to perform scale-up. We can look at different ways of growing cells, but fundamentally the methodology is utilizing putting cells onto a non-porous 2-dimensional surface, and we utilize generally serum as a supplement for culturing the cells. Serum in and of itself is relatively non-physiologic. Serum is plasma with the clotting factors removed, so the only time that cells will see or be exposed to serum is when they're in an activated state. And the growth of cells on a 2-dimensional non-porous support means that we must maintain cells in a monolayer configuration, and this is also a non-physiologic way of growing cells. When we go to scale up our cell culture, we can look at things like roller bottles, cell factory, cell cube. These are all just ways of getting more surface area into a certain amount of space. And other options for cell culture scale-up will include bags, spinner flasks, bioreactors such as a 50-liter bioreactor that's pictured here. This is also a non-physiologic way of growing cells at low density, 1 to 5 times 10 to the 6 cells per mil. In a suspension type culture, we have to put surfactants into the media to protect the cell membranes from the shear, so the cells are exposed to a lot of different physical shear forces as well. All of these methods, to a greater or lesser degree, are going to subject the cells to what we call a feast or famine, which is a non-homeostatic process of growing cells in which the cell culture conditions are constantly changing. So we're familiar with, of course, lag phase. Even though we put in the cells, we feed the cells into a flask, they still take a little bit of time to condition their cell environment around them with their own cytokines and growth factors. So it takes a little while for the cells, even in the presence of serum and on plastic, to begin to proliferate. Then we have our optimum cell culture conditions, and here we define optimal cell culture conditions as being that which gives us the best monoclonal antibody production, but I utilize this as a monitor for all types of cell physiology and cell health. And just when things are getting good, it's time to split the cells because they've reached confluence, and we must maintain them in a monolayer subconfluent fashion, because in flasks, nutrients come from the top down, nutrients and oxygen come from the top down, so if the cells pile up on top of one another, they block the delivery of nutrients and oxygen to the bottom layer of cells, and everything dies, peels off, and floats away. So it's important then to understand that the growth of cells on a non-porous two-dimensional plastic surface in the presence of serum is an inherently non-physiologic process, although still has been a valuable tool for use in research and has certainly have widespread utilization all around the world. But wouldn't it be nice if we could maintain the cells at their more homeostatic conditions? Wouldn't it be nice if we didn't have to split the cells, if we didn't have to digest them every 2 days and move them to a new plastic dish? It'd be nice if we could maintain them in their most homeostatic cell culture environment for extended periods of time. And in point of fact, that's what we do with a hollow fiber bioreactor. It is mimicking the human circulatory system. And in fact, the world record for continuous culture of a glioma cell line in a hollow fiber bioreactor is 2 years of continuous culture and continuous production of a glioma cell line. In one of our cartridges, we typically can culture hybridomas for as long as 6 months or even up to a year of continuous culture, continuous production with no change in antibody affinity, avidity, or glycosylation patterns. CHOs and 293s we can typically grow for 3 to 6 months worth of continuous production. So hollow fiber cell culture is based on the principle that each one of these fibers inside the cartridge is like a little filter, and it's shaped like a drinking straw, 200 microns in diameter. Inside the cartridge there are literally thousands of these fibers, about 4,000 to 7,000 fibers. We seal the fibers up in the end in a medical grade polyurethane and then trim off the ends so that only the open ends of the fibers are available to the end port of the cartridge. So any media that is pumped through the insides of the fiber necessarily goes through the insides of the fiber, and typically we grow the cells on the outside of the fiber. So what we've done is created a semi-permeable barrier of defined molecular weight cutoff between the area in which the media is flowing and the cells are growing. This differs from traditional flask culture in 3 important and fundamentally different ways. The first is that we have a tremendous amount of surface area in a really small space. And as I mentioned earlier, biology is all about surface area to volume ratio. So our standard cartridge, the C2011 cartridge, has 3,000 square centimeters of surface area and a volume of 20 mils. So we have 150 square centimeters of surface area per milliliter volume. Our larger cartridge, the C2018, has 1.2 square meters of surface area and a volume of 60 mils. So we have an even higher surface area to volume ratio. We have 200 square centimeters of surface area per milliliter volume. So you can imagine a film that's 50 microns thick laid out on a flat plastic plate. This is the kind of surface area to volume ratio that we have. This is what allows us to grow cells at extremely high densities, at 1 to 2 times 10 to the 8 cells per mil. It is the only way to culture cells and to maintain cells at in vivo-like cell densities. Here in the photo you can see these are actually lymphocytes growing at approximately 1 to 2 times 10 to the 8 cells per mil, tissue-like cell density. Secondly, the cells are bound to a porous support, not a non-porous plastic dish. The nutrients come from the bottom up, not from the top down. This is what allows us to culture the cells for extremely long periods of time. This is what allows us to grow cells in 3 dimensions. Passage number is irrelevant, and splitting the cells is not required. Thirdly, we can control the pore size of the fiber, anywhere from 5,000 Daltons up to 0.1 microns. So the idea is that small things like glucose, lactate, ammonia, nutrients and waste products can freely cross the fiber. Larger things that are secreted by the cells, such as proteins, antibodies, viruses, exosomes, cannot cross the fiber. So they'll be retained by the fiber and concentrated to a very high degree, anywhere from 50 to 100 times higher concentrations than what you can get out of standard bioreactors. It's important when you're dealing with hollow fiber to understand exactly what a molecular weight cutoff is. So we have a 5,000 Dalton and a 20,000 Dalton fiber. It's important to remember then that the Dalton is a fundamental unit of measurement of mass, and a Dalton is equivalent to the mass of a hydrogen atom. It was defined by Sir Joseph Dalton in 1786 at Cambridge University in the UK. So a Dalton is the mass of a hydrogen atom, and the hydrogen atom is the smallest intact atomic particle that you can have before you start going subatomic. Nowadays, NIST likes to define the Dalton as 1/12 the mass of a carbon atom. because they like to relate everything to carbon, but it's fundamentally the same thing. So we can then understand that the average mass, the average molecular weight of an amino acid is around 120, and we could take something like an antibody, an IgG1, and its molecular weight is 144,000 Daltons. So by this, then we can understand that an IgG1, an antibody, will have approximately 1,200 amino acids making it up. So these three things, the high surface area to volume ratio, the fact the cells are bound to a porous support, and the ability to control the environment around the cells and to concentrate their secreted products, are the three factors that are fundamentally different and represent a unique cell culture environment versus flash culture and other types of bioreactor methods. So as I mentioned, this is just a picture of the cells at high density. We're looking at 10 to the 8th plus cells per mil. In our particular system, we utilize wavy fibers, so the fiber bundle pushes against each other inside the cartridge, so they're uniformly distributed inside the cartridge shell. So these cartridges are optimized for both suspension and adherent cells. It's this high cell density that permits adaptation to lower serum concentrations and to our protein-free media that we've developed specifically for hollow fiber bioreactor culture, CDM-HD, and we'll talk about that a little bit later. So the system itself is a closed, biosafe system. We have a central reservoir that contains the media, and the media recirculates through the cartridge. We utilize a pumping system. We actually have a specific positive pressure displacement pump that we've developed for this system. We call it a squishy pump. It's not a peristaltic pump because peristaltic pumps are friction-based and will shed plastic off the pump tubing and clog up the fiber bundle. And also, it's very difficult to utilize a peristaltic pump to generate 80 to 160 mils a minute recirculation rate, which is what we use with the hollow fiber system. In any bioreactor, the rate limiting factor is generally not the delivery of nutrients and removal of waste products, but the delivery of oxygen and removal of CO2. So we utilize a loop of thin-walled silicone tubing. The standard cartridge has 2 meters of this tubing to provide oxygen exchange and CO2 removal. It's very highly gas permeable. So that's where the gas exchange takes place, so the media just continuously recirculates, and we just simply measure the glucose that's present in the media in the central reservoir and harvest our product on a schedule based upon our protocols. So here at where I'm based at in Frederick, we have US Amrit just down the road, and they're very much involved in different types of interesting biology, and we had some protocols and discussions with them in the past about producing a virus, and they called me up once and they said, oh, we'd like to produce a virus. And I said, okay, what kind of virus do you want to produce? And they said, well, we can't tell you that. And I said, okay. I said, what kind of cells do you want to grow the virus in? And I said, oh, African green monkey kidney cells. And I said, oh, okay. So you want to grow Ebola? And they laughed, and they said, no, no, no. They said, time to burst with Ebola, i.e. the time from when a virus hits the cell and the cell bursts, time to burst with Ebola is 12 hours, and time to burst with our virus is 4 hours, so it's actually 3 times as virulent as Ebola, and they definitely appreciated the fact that this was a closed, biosafe system. So this is just a breakout of how the cartridge is put together. You can see the loop of silicone tubing in the back, and we have 2 one-way check valves that works just like your heart. And then our pump piston will squeeze on this pump tubing that you can see coming up from the in the back of the flow path. So it squeezes on that tubing and utilizes the one-way check valves to generate the flow rate. And you can see the loop of silicone tubing where the gas exchange takes place. So the system is designed to fit inside any standard-sized incubator. Temperature is controlled by the incubator. Gas is controlled by the incubator. It's a very simple system without any kind of fancy sensors or anything like that. It's all going to be measured manually. And we utilize a thin cord for power, so we don't have to worry about having a plug inside the incubator. And it's very convenient to put inside the incubator and to take out again. So when working with the cartridge, we can pull the cartridge out of the Duet pump and move it into the hood. So it's both the easiest and the most difficult process cartridge to contaminate. While it's outside of the hood, it's going to be sealed. When it's inside the hood, it's very important to utilize good sterile technique. Good sterile technique is always a plus. Maintenance is only 15 minutes a day. We want to just measure the glucose and harvest our product. As we mentioned, good sterile technique is always a plus, and here in this particular example, it's important to notice this nice young lady happens to not be wearing a lab coat. So we want to be sure that we follow good sterile technique whenever we're working with the cartridge. So we're going to measure the glucose, and the glucose measurement is very important. It tells us directly what's going on inside the cartridge. The total amount of glucose in the media tells us when it's time to change the bottle. All we have to do is unscrew the bottle and put on a fresh one. So generally we'll change the media before the pH is shifted, and we'll change it when the glucose is about 50% depleted. Typically we'll work with DMEM, and starting glucose on DMEM is 4.5 grams per liter. So when it gets down to about 2 grams per liter, it's time to change the media. So we just unscrew the bottle and put on a fresh one. The glucose uptake rate, or the amount of glucose consumed in a 24-hour period of time, is a direct indicator of how many cells we have in the cartridge, how happy they are, how healthy they are, what's going on. As long as the glucose uptake rate is either plateauing or increasing, everything's good inside the cartridge. If we see that it drops, that means that something is amiss inside our cartridge, and we want to take a look at what's going on. The glucose uptake rate for this cartridge will typically be in the range of 1 to 1.5 grams per day. This is going to be directly representative of about 1 times 10 to the 9th cells. So the applications that we'll talk about today, certainly monoclonal antibody production. Hybridoma culture is one of the basic raison d'etres for hollow fiber when it was developed back in the early 80s for commercial applications. We'll go into that in a little bit more detail in a second. Recombinant protein production. It is certainly a way of producing anywhere from 10 milligrams on up to gram quantities of recombinant protein from a mammalian expression system in small volumes. Conditioned medium, of course. We have a lot of exciting applications now for exosomes. It is a physiologically relevant way of culturing endothelial cells under shear stress, and cell co-cultivation and other types of cell culture models. And also very important that we're excited to be involved with, but I won't get into too much detail for this particular audience, is going to be the use of the system for antibiotic pharmacokinetics and pharmacodynamics for determining resistance generation and best dosage profiles, and total kill and 2-drug models for antibiotic applications, which is of course going to be quite critical these days. So this is a cartridge. This is going to show the kind of cell densities that we can achieve. It's a little bit of an extreme picture of it, but we can see the cells that collect in the ends of the cartridge. This is because the diameter of the cartridge expands slightly at the end so that we don't have the potting material running up the sides of the cartridge due to capillary action. But we can see that that density is uniform really throughout the entire cartridge. It's just more visible at the ends. But this is what cells look like when they're growing at 10 to the 8 cells per mil. So the advantages overall for hollow fiber cell culture is our product is going to be more concentrated, generally 50 to 100 times more concentrated than what you'll see out of regular standard flask or spinner culture. We will demonstrate uniform and complete post-translational modifications over extended periods of time. I believe this is due to the improved and more physiologic cell culture environment that the cells are growing in. We also see significantly less apoptosis. May not be exactly the right definition of the word. What we see is low rates of cell lysis. So it's really surprising to me when we work with this that we'll see lots of dead cells as part of the growth process, but very little cell fragments, less contamination with intercellular proteins and DNA and membrane fragments and these kinds of things. And we'll show the consistency of this production over many months. And then, of course, we have developed our own in-house CDM-HD, chemically defined media for high-density culture. You don't have to use CDM-HD with our system. You can work with just about any media that you like, although we find that the more expensive a media is, the more complex a media it is, the less well it seems to work with our system. So CDM-HD, to me, is not just a protein-free, chemically defined, animal component-free, CGMP-compliant serum replacement. It has actually been simplified and modified and optimized for use in our hollow fiber bioreactor. And to me, it's really more a manifestation, a direct physical manifestation of the different cell culture environment found in a hollow fiber bioreactor, because CDM-HD does not work in flask culture. CDM-HD does not work in spinner culture. The cells need to be at higher density before we can support them with CDM-HD. So typically we'll culture the cells and inoculate the cells at low density in the cartridge with serum, and then as the cells grow, we get them up to high density, we can simply switch over to CDM-HD. It really doesn't even require any adaptation. Kind of surprising. So as I said, it's optimized and simplified for the hollow fiber bioreactor. Importantly also, since the cells do not experience any shear within the system, there's no requirement for surfactants to protect the cell membrane from shear. It's chemically defined, protein-free. I'm also kind of surprised by the number of components. There's about 92 different components in CDM-HD. It has some heaps to stabilize the pH. It has a little bit of extra glucose. It's got a lot of amino acids in there. If you want to make protein, you need amino acids. It has micronutrients. It has all kinds of things in there. And it is CGMP compliant. If you do want to utilize it in a CGMP compliant process, we can, with the proper paperwork and NDA in place, provide you with information on the components that are contained within. So we can get very high lot-to-lot consistency. We can ship this at ambient, store it at 4 degrees, 3-year shelf life on CDM-HD from date of manufacture.